Hello everyone and welcome to today's session. Today we'll be discussing the NCERT Exemplar questions for the chapter Morphology of the Flowering Plants. This is the part 2 of the lecture. Before starting this lecture, I just want to give you one information that the images, text and other materials used in the presentations are for education purposes only. We respect the intellectual property rights of the original creators and owners. If you are the owner and have any concerns or issues, please contact us at sathee@itk.ac.in and we shall remove the content immediately. Please note that parts of the content may have been generated using generative AI, which is experimental. So what are the objectives of today's session? So we will practice, we will understand, and we will master the basic concepts, okay, by discussing NCERT exemplar questions. So these questions, they are based on certain concepts. We will understand this concept by working through NCERT exemplar questions. So here you can see these questions are divided into these categories. So there will be multiple choice questions and there will be questions like match the column. There are very short answer type questions, short answer type questions, long answer type questions, and total questions are 16. So let's start with this question. This is a multiple choice question. The question is endosperm. A product of double fertilization in angiosperm is absent in the seeds of. So endosperm, this is found by triple fusion. Okay, in the case of angiosperms, where double fertilization occur, this angiosperm is absent in seeds of which plant? You can get the answer of this question from the NCERT section 5.7. Let's have a look at the options here. Option B is coconut, B is orchid, C is maize, and D is castor. What would be the correct answer here? So let's have a look at the answer. So answer here will be option B, orchids. Let's have a look at the explanation now. Orchids, these seeds are non-endospermic, means the endosperm is absent here. Okay, endosperm, as you know, they nourish the developing embryo. Okay, so there's no endosperm in the orchids. Usually endosperm is found before the embryo because it provides nourishment to the embryo. The endosperm, this is absent, as during seed development the endosperm is completely used. Okay, and nourishment is then provided by the food which is present in the cotyledon. So nourishment of the embryo, this can be provided by endosperms or the cotyledons. Cotyledons also store the seeds. So these cotyledons, they are also known as seed leaves. Now all other options are endospermic seeds. Now let's discuss this another question. The question is, many pulses of daily use belong to one of the families below. Okay. So what is the family to which the pulses belong? So for more information on this question, you can read the NCERT section 5.9. Options are Solanaceae, Fabaceae, Liliaceae, and Poaceae. What will be the correct answer here? Okay, so let's have a look at the answer now. Answer is option number B, which is Fabaceae. The Fabaceae is a subfamily of Leguminosae, which was earlier called Papilionoideae. Now, plants of this family are source of pulses and edible oil as well. Now, pulses are rich in protein contents. The next question, the placenta is attached to the developing seed near the. What are the options here? Testa, hilum, micropyle, and the chalaza. What will be the correct answer here? Let's have a look at the answer. So, answer is hilum. Hilum is also known as seed scar. Let's have a look at the explanation now. So, placenta is a special tissue by which ovule is attached to the ovary. Okay, it has a special kind of arrangement in the ovary which is called as placentation. Now, the placenta is attached to the developing seed near the hilum. The hilum is also known as the seed scar. So, it is a scar where the seed breaks from the funiculus. Funiculus is the stalk of the ovule. Okay. So, hilum is a scar where the seed breaks from the funiculus, connecting seed with the fruit wall. So, hilum represents a scar, okay, which is connecting seed with the fruit wall. Now, rest of the options are incorrect. So, here you can see this is a detailed structure of anatropous ovule showing chalazal, hilum, and the micropyle. So, this is the chalazal end, okay, and the lower one, this is the micropylar end. Okay, you can see the funiculus here, funicle, which is the stalk of the ovule. You can see the embryo sac which is present within this ovule. Okay, then micropyle, this is a region where the integuments are absent. There are two integuments here which later on develop into the seed coat. Clear? Then you can see this is the hilum here. So hilum, this is a scar where seed breaks from the funiculus and it connects the seed with the fruit wall. So ovule develops into seeds later after fertilization. Clear? So it connects the seed with the fruit wall. Now testa, this is the outer seed covering. So integuments, they develop into the seed coat, also known as the testa. Micropyle, what is the function of the micropyle? So through micropyle, the oxygen and water enters during the seed germination. And chalazal end, it joins nucellus and the integuments. So at the chalazal end, antipodals are present. Okay, nutrients travel through funiculus and integuments via chalaza into the nucellus. Okay, so again you can see the vascular bundles are also present here within the funiculus. And with the help of these vascular bundles, nutrients travel through the funiculus and integuments via chalaza into the nucellus. Next question, which of the following plants is used to extract the blue dye? So blue dye is extracted from which plant? So what are the options here? Trifolium, Indicofera, lupin or cassia? What will be the correct answer here? Let's have a look at the answer now. the answer is option number B, Indigofera. Let's have a look at the explanation. Okay. So first of all, you have to keep this thing in mind. Dyes, they are the secondary metabolites of certain plants and are very important economically. So plants, they produce a lot of secondary metabolites. These secondary metabolites, sometimes they have bioactivity also. Okay, and they are the byproducts of the metabolism. So dyes, they are nothing but they are secondary metabolites. So they are chemicals produced by the plants. Okay, and they are very important economically. So they have economical importance. Okay, so answer is Indigo tinctoria. Okay, so this blue dye is obtained from Indigo tinctoria and from Saffrutigo plant as well, which belong to the Fabaceae family. And these plants, they produce blue indigo dye. Other options are incorrect. Trifolium is used as a fodder. Lupin is an ornamental plant. And Cassia, this is a shrub usually grown on the roadside as an ornamental plant. Now let's have a look at this question. So this question is column-based question here. Okay, here two columns are given to you, column 1 and column 2. You have to match the following columns. Okay, so this type of question you can get in NEET exam as well. So what is given in column 1? Aileron layer, parthenocarpic fruit, ovule and endosperm. And in column 2, again some terms are given to you like nutrition without fertilization, seed and double fertilization. You have to match the column 1 with the words given in the column 2. Okay. Now 4 options are given to you. What will be the correct answer here? From the options A, B, C and D. Let's have a look at the answer now. The correct answer is option number A. So aileron layer, this provide nutrition. Okay, aileron layer differentiates the endosperm from the embryo, and endosperm is surrounded by this aileron layer. This consists of proteins. So aileron layer, this surrounds the tissue of monocot seed and is distinct from the seed. Okay, so aileron layer is present outside the endosperm. It is made up of protein and this is present in monocot seeds. It is a proteinaceous layer which provides nutrition and helps in the germination, seed germination as well. Now, parthenocarpic fruits, they develop without the fertilization. Okay. Parthenocarpic fruits, they are seedless. They develop without the fertilization, and there is a hormone auxin that can also induce parthenocarpy in the plants. Okay, because there is no fertilization, so no seeds are produced here. Now, the ovule contains the female reproductive unit that develops into the seed. So, after the fertilization, ovule develops into seeds. And then endosperm. This is formed by triple fusion. Clear? Which is one of the fertilization in the double fertilization. So what happens in double fertilization? This is a characteristic feature of angiosperm, which are the flowering plants. And in double fertilization, first of all, syngamy occurs. In syngamy, what happens? One of the male gamete fuses with the egg. This is known as syngamy. This results in the formation of a diploid zygote. Zygote is a unicellular diploid structure. Then Next one is the triple fusion, where another male gamete fuses with the two polar nuclei. This results in the formation of primary endosperm nucleus, which is triploid. Okay, and the central cell finally transform into the endosperm, which consists of the primary endosperm nucleus. Okay, so that's why option number A is the correct answer. A matches with 1, B matches with 2, C matches with 3, and D matches with 4. Now let's discuss some very short answer type questions. So here fill in the blank is given to you. You have to fill the blanks here. So question is, reticulate and parallel venation are characteristic feature of dash and dash respectively. Okay. So reticulate venation is observed in which plants and parallel venation is observed in which plants? In reticulate venation, what happens? There is a network of veins. In parallel venation, the veins run parallel to each other. What is the correct answer here? The correct answer here is reticulate venation is a characteristic feature of dicot and parallel venation is that of monocots. So all dicots have reticulate venation. That is, veins they form the network, except in alocasia and smilex. These are exceptions. In monocots, parallel venation is observed, means veins run parallel to each other within the lamina. This is also known as blade. And there is an exception, calophyllum. Okay, so you should remember the exceptions as well. The next question, in epigynous flowers, ovary is situated below the. Okay, we are talking about epigynous flowers. So there are perigynous, hypogynous, and epigynous flower. So ovary, they are asking about the location of the ovary. Ovary is situated below the. Let's have a look at the answer now. So in epigynous flower, ovary is situated below the thalamus. So ovary is inferior here. Flower is superior. Now other whorls of flower like sepals, petals, and androecium grows above the ovary. So ovary, you can see here is, you can say superior because it is situated below the thalamus. Clear? So it is inferior. Example is carrot, guava, cucurbita, flower, etc. Sunflower, etc. So this is image showing you epigynous flower here. Okay. So here you can see all the parts of the flowers. Okay, they are showing you the ovary, ovule, and the thalamus as well. So here the ovary is situated below the thalamus. Now next question, you have to add the missing floral organs of the given floral formula of the Fabaceae. Okay, so this is a floral formula that is given to you. Floral formula of Fabaceae. You have to add the missing floral organs. What are floral organs? Calyx, corolla, androecium, and gynoecium. Let's have a look at the answer now. Okay, the floral formula of Fabaceae family is, okay, it is written like this. So here missing was C1 plus 2 plus 2. Okay. Sometimes people write it as C5 as well, but this is the correct way to write is. So this corolla was missing. The Fabaceae flowers are bisexual and they are zygomorphic. So for the zygomorphic, this sign is used. Okay, like percentage. Then for bisexual, both male and female parts are present in the same flower. Okay, then you have calyx 5, then corolla, then androecium 9 plus 1, and then gynoecium. Okay, it has 5 fused sepals. The gamocypress condition is present here. Consists of 5 petals forming standard wings and keel. That's why 1 plus 2 plus 2. Okay, so this is the better way of writing C1 plus 2 plus 2 because the petals, they are of 3 types here: standard, wing, and keel. That's why C1 plus 2 plus 2. It has 10 diadelphous stamens. That why 9 plus 1. Okay, so these stamens, they are in groups of 2. Okay, diadelphous means in groups of 2. And there are 10. So that's why 9 plus 1. There are 2 groups of the stamens. And in total, there are 10 stamens. And the ovary is superior ovary with single carpel. So G1, and you are underlining it. Okay. Now, next question: name the body part modified for food storage in the following. Okay, so carrot, sweet potato, radish, dahlia, gladiolus, portulaca, colocasia, asparagus, potato, turmeric, and ginger. What will be the answer here? So you have to name the body part which is modified for food storage here. Let's have a look at the answer now. So for carrot, taproot modification, sweet potato is a root modification, radish is also a root, colocasia, stem, which is known as corm here, asparagus, root, and potato, this is the modification of the stem. Stem is modified to store the food. Okay, so these are the correct answers here. Okay, so here you can see in potato, for example, stem is storing the food here. In asparagus, root is storing the food. Then in dahlia, these are adventitious roots. What are adventitious roots? Roots which arise from any other region other than the radicle. Then a gladiolus stem, in portulaca, adventitious root, in turmeric stem, and in ginger, again it is modification of the stem. Because in ginger you can see nodes and internodes as well. So wherever you see nodes and internodes, these are stem. Turmeric also stem. Stem is storing the food. The next question is short answer type question. Now the question is reticulate venation is found in dicot leaves, while in monocot leaves venation is of parallel type. Now biology being a science of exceptions, find out an exception to this generalization. So you have to list some exceptions. Okay. So what will be the correct answer here? So usually in dicots, reticulate venation is seen, and in monocots, parallel venation is seen. So what are the exceptions to this generalized observation? Let's have a look at the answer now. So reticulate venation is the characteristic of dichots and parallel venation is of monochots. This is the generalized knowledge that we have. But few exceptions are also seen in this generalization. Parallel venation is also found in dicot plants. So in some dicot plant, parallel venation is also seen. Example is Carophyllum and Corymbium. Okay, so these are the dicot plants where parallel venation was observed. Now, reticulate venation is also found in monocot plants such as Alocasia and Smilex. So these both are monocot plants, but they have reticulate venation. So remember this. The next question is, you have heard about several insectivorous plants. Okay, so these plants, they feed on insects, they obtain nutrition from the insects, especially nitrogen. Okay, Nepenthes or pitcher plant is one such example. Nepenthes khasiana is the full scientific name. So this plant grows in shallow water or in marshlands. Now, what part of the plant is modified into pitcher? And how does this modification help the plant for food, even though it can photosynthesize like any other green plant? What will be the correct answer here? Let's have a look at the answer. So, Nepenthes khasiana, this is an insectivorous plant. Here, the leaf lamina modified into pitcher, and the petiole coils like a tendril. Okay, so it is kind of modification of leaf, you can say. And the posterior part of the petiole remains flattened, and the apex form a lid. And digestive enzymes in the pitcher digest the trapped insects. So the pitcher, this is the modification of the leaf lamina. It becomes pitcher-like. Now these adaptations develop to overcome nitrogen deficiency in the marshy soils. So basically, which nutrients are they absorbing from these insects? So they are basically absorbing nitrogen from these insects. Okay, because this nitrogen, there is a lack of nitrogen in the marshy soil. So this is the kind of adaptation, okay, to fulfill this nitrogen deficiency. So here you can see this is image showing you the Nepenthes khasiana, also known as the pitcher plant. Here you can see the pitcher shaped leaf. So leaf is, this is the kind of leaf modification. Okay, this picture is shown to you here. And the petiole also develops into tendril like. So this is tendrilar petiole. Okay, so petiole also modifies to become tendril like. And the leaf lamina, leaf lamina is also known as leaf blade. This modifies into pitcher. And when the insects come, okay, and this lid closes and the insects are digested with the help of digestive enzymes. The next question, mango and coconut are drupe type of fruits. So there are many types of fruits, berries, drupes. Okay, so mango and coconut, they both are drupes. In mango, the fleshy mesocarp is edible. Which is edible part of the coconut? And what does milk of tender coconut represents? What will be the answer here? Let's have a look at the answer. So these coconut, they have the fibrous mesocarp. Mango have the fleshy mesocarp. So mango and coconut both are drupe fruits, and they develop from monocarpellary superior ovaries. Mono means one. So there's only one carpel. So they develop from monocarpellary superior ovaries and are one-seeded. Now it is differentiated into outer thin epicarp, middle fleshy mesocarp, and inner stony endocarp. Okay. So the fruit wall is divided into epicarp, mesocarp, and endocarp. Fruit wall is known as the pericarp. Okay. So in case of mango, it is fleshy mesocarp. In the case of coconut, it is fibrous mesocarp. So you can see here, these are the parts of the fruit. Image A is showing you the mango. Image B is showing you the coconut. You can see there is a fleshy mesocarp in the case of mango, fibrous in the case of coconut. They have only one seed. You can see the endocarp as well, and the epicarp is also visible. And the what does the milk of tender coconut represent? So basically, it is what? Let's have a look at this. Adipal part of the coconut, or what is the scientific name of coconut? Cocos nucifera. This is endosperm. Okay. So initially there is a liquid endosperm, and then after cellularization of the liquid endosperm, it becomes solid. Okay. Now milk of tender coconut represents the liquid endosperm. Okay, so endosperm initially is in liquid form because there is no cellularization. There is no cell wall formation here. After cellularization, it becomes solid. Now later it gets deposited along the walls of the endocarp and forms edible flesh after cellularization. The next question, how does you differentiate between free central and exile placentation? So this question is related to type of placentation. Okay, so how will you differentiate between free central and exile placentation? Let's have a look at the answer. So what is placentation? Placentation is the arrangement of the ovule on the walls of the ovary with the help of special kind of tissue called the placenta. Now plants show different type of placentation, and central and exile placentation are among them. Now they have the following differences. What are the difference? So this is a table showing you difference between free central placentation and exile placentation. Now first, in free central placentation, ovary has single chamber. Okay, means it is unilocular. Exile placentation, ovary is multi-chambered. It is multilocular. This is first difference. Next, in free central placentation, ovules are present on the central axis, free inside the ovary, and there is no septa. In the case of exile placentation, placenta is on the central axis, where septa fuse to form the exile column. You should remember the example also. Example of free central placentation is dianthus and silenza. An example of exile placentation is citrus and two. Now next question, the arrangement of ovules within the ovary is known as placentation. Now what does the term placenta refer to? You have to name and draw various type of placentation in the flower as seen in transverse section or the vertical section. Let's have a look at the answer. So placenta, this is a flattened cushion-like tissue on which one or more ovules are attached. Okay, so this is what is placenta. Now the various type of placentation seen in flowers are described in the following table. So let's have a look at the different placentation types. So you can see first one is the marginal placentation. There is a single placenta which develops along the junction of two fused margins as seen in P. So this is marginal placentation, you can see. Okay. Then you can see axile placentation here. What are the feature of axile placentation? There is a multicarpellary. Okay, means many carpels are present. Syncarpus means carpels are fused together. Syncarpus gynoecium. Inward growth of the carpus margin forms a multicarpellary condition. Okay, that contains and exists in the center. Okay, where the ovules are born. Placenta arises from the central axis bearing the ovules. So placenta is in the arises from the central axis and this is bearing the ovules. Example is solanum and China rose. Now let's talk about the parietal placentation. The number of placenta correspond to the number of fusing carpels here. Sometimes the ovule bearing placenta grow inwards to form the false septa as well. Okay, but you can see here, sometimes it forms the false septa. Usually there is no septa here. Okay, when if there is a false septa, then the ovary becomes bilocular, but usually it is without any locule, so it is unilocular. Okay. Example is mustard, sometimes where false septa can be present, or sometimes it can be trilocular as well. Example is cucurbita. Then free central placentation, ovary is unilocular, ovules are born on the central axis again. Okay. But there is a difference between axile and free central placentation because here there is no septa, you can see. Okay, ovules are born on central axis, not connected to the ovary wall by a septum, so there are no septum here. Example is dianthus and silenza. Basal, you can see basal in basal placentation, you have unilocular ovary. Okay. The ovary is unilocular and the single ovule is born at the base of the ovary. You can see single ovule is born at the base of the ovary attached to a single placenta. Example is sunflower and wheat. Now we have long answer type question. What is the question here now? Sunflower is not a flower. Explain. So is sunflower a flower? Or is it combination of flowers? Or is it an inflorescence? You have to explain it. Let's have a look at the answer. So sunflower is not a flower, but it is a kind of inflorescence called capitulum. So capitulum is the right answer here. Okay. So what are the features of the capitulum? Capitulum, in the capitulum, the receptacle is flattened. So another name of thalamus is receptacle. It bears numerous sessile and small florets. Okay, so these florets, they are sessile. They are without the stalk. There are two kinds of florets, ray florets and disc florets. And the youngest florets is in the center, and the oldest florets lies at the periphery. Now, whole cluster of florets gets surrounded by the bracts, which is known as the involucre. Okay. Now, two kinds of florets are recognized in the sunflower. Okay. First one is the ray florets. So we've discussed there are two florets in a sunflower. Okay, that's why it is an inflorescence. It is not a flower. Okay, so there are different kinds of inflorescence. There is spike of spikelets, then there is capitulum, verticillaster. Okay, you should remember this verticillaster, this capitulum, spike of spikelets. So these are the important inflorescence. Okay, basically these inflorescence are divided into two main categories, racemose and cymose. So these are the modification of cymose and racemose inflorescences. Ray floret, they are arranged on the rim of the receptacle, having distinct yellow and strap-shaped petals. Okay. Then these florets are female, but they are sterile and are always zygomorphic and may be arranged in one or more whorls. So ray floret, they are yellow. Okay, they have yellow petals. They are sterile, they are female. Now, what about disc florets? So they are grouped in the center. Okay. They are bisexual, means they consist of both the male and the female reproductive organs, and they are actinomorphic. So again, a question can come here. What are the difference between ray florets and the disc florets? So here you can see this is the capitulum of sunflower with disc and the ray floret. Sunflower, this is the capitulum inflorescence. Here the lateral section of inflorescence is shown to you. Here you can see disc florets are present. They are grouped in the center, and at the periphery, these ray florets are present. And these ray florets can be present in one or more whorls. Then you can see this is a disc floret, and ray florets are also shown to you. Ray florets, they are female and they are sterile. These are yellow in color. And disc florets, you can see they are bisexual. They consist of anthers also. The anther is a part of stamen. Stamen consists of filament and anther. And style and stigma are also seen here, which are the part of the carpel. Ovary, style and stigma, they are, this together forms the carpel. So disc florets, they are bisexual. They consist of both male and the female reproductive structures. These are actinomorphic as well. Ray florets, they are zygomorphic. The next question, how do you distinguish between hypogeal germination and epigeal germination? So there are two types of germination, hypogeal, epigeal. Now, what is the role of cotyledons and the endosperm in germination of these seeds? Okay. So, what would be the answer here? Let's have a look at the answer. Now, the difference between hypogeal germination and epigeal germination is as follows. So, hypogeal seed germination, there is a rapid growth and elongation of the epicotyle. And in epigeal seed germination, there is a rapid growth and elongation of the hypocotyle. This is the major difference. Okay. Epicotyle is the region above cotyledon, hypocotyle is the region below the cotyledon. So, in hypogeal germination, there is a rapid elongation of the epicotyle. In epigeal, there is a rapid elongation of the hypocotyle. Now, in hypogeal germination, cotyledon remain inside the soil. Cotyledon, they are also known as the seed leaves. In epigeal germination, the cotyledons emerge above the soil level because of rapid elongation of the hypocotyle. Because of this, the seed leaves, cotyledons, they emerge above the soil. But in hypogeal germination, they are below the soil. Here in hypogeal germination, as they remain inside the soil, they are non-green, they are non-photosynthetic. But in epigeal germination, as they are above the soil, they become green and photosynthetic. Okay. Then you should remember the example of hypogeal germination, castor and beans, the example of hypogeal germination. And in maize and rice, epigeal germination is observed. Okay, this is clear. Here in the image, you can see first image is that of hypogeal germination. Here the cotyledons remain inside the soil. Okay, and here you can see there is a rapid elongation of the epicotyle. Epicotyle will later form the stimule. Then you can see in the epigeal germination, the cotyledons, they emerge out of the soil. So here these cotyledons, you can see they have emerged out of the soil due to rapid elongation of the hypocotyle. Okay, so these are the major differences between hypogeal germination and epigeal germination. Now, what are the role of cotyledons and endosperm? So both of these, they store the food. They provide nourishment to the embryo. Okay, so cotyledons and endosperm, they contain the reserve food materials. And these reserve food materials, they are consumed by the embryo. Okay, these endosperms, they develop before the embryo. Okay, so they are formed before the embryo. Endosperm development occurs before the embryo development. Now, when seeds imbibes the water, so for seed germination, water is very important. And this water and oxygen, this enters through the micropyle. Okay, micropyle is a region where the integuments are absent. So whenever the seed imbibes the water, okay, we will write imbibe here. Then what happens? The enzymes get activated, such as amylase enzyme. And this amylase enzyme, they act on carbohydrates. So these enzymes get activated and then they hydrolyze the reserve food material, which is present in the endosperm, okay, as well as the cotyledons. So both of these store reserve food materials such as carbohydrates. So basically they store carbohydrates. That's why amylases are usually activated. And after the hydrolysis of the, you can say, carbohydrate or the reserve food material, Then this food, which is broken down now, this is made available for the seed germination. So initially what you see, what happens, the seeds, they are, you can say, whenever the seeds, they are bored in the soil, they are present inside the soil. So seeds cannot photosynthesize. So how the seed germination occurs without photosynthesis? How the embryo can survive without photosynthesis? So initially the seed have the reserve food material inside it in the form of endosperm and the cotyledons. Cotyledons, they are also known as the seed leaves. Okay, and then later the seed germinate and the seedling is formed. Okay, and then the plant can photosynthesize. Sometimes the cotyledon also start photosynthesizing, as we have seen in the case of epigeal germination. Okay, so this is the role of cotyledons and endosperm. So this is a very important question actually. This question can come. Difference between hypogeal germination and epigeal germination. Question can come in the NEET exam as well. Okay, you should know the examples also of hypogeal germination and epigeal germination. And you should know about the exceptions also that we have discussed today. So we have discussed the exceptions of the parallel venation and the reticular venation as well. You should know that also. And whenever you are writing the answers for any question, draw the diagrams and explain the concept using diagrams and examples. Okay, so this will provide clarity to whosoever is analyzing that answer. Okay, so we have discussed the exemplar as well, related to morphology of the flowering plants. Okay. So this is the end of this session. I hope this session was quite knowledgeable for you. We'll meet you in the next session. Thank you very much for listening.